Thanks, Mike. Really happy to be here and happy to get to tell you a bit about this Bioconductor Africa initiative that we started running recently. So to get straight to it, like why Africa and why now? Main reason, we started getting requests to have Bioconductor in-person workshops held in Africa. We could see Africa was underrepresented in our map of users. So this world map, the red dots representing Bioconductor users around the world. And you can see there's not that many red dots in the Africa region. However, Africa, almost 1/5 of the world's population lives there. So we recognized that they had been underserved for genomics and Bioconductor training. So to talk briefly a bit about Bioconductor training. So there's a lot of resources in the training ecosystem. It ranges from package help functions to vignettes to workflows, which there's a BOF on at 5 o'clock on that topic. There's books on things like single cell analysis, high-C, spatial transcriptomics, so more detailed analyses than what you find in individual package vignettes. And then we also have workshops, so from a few hours, like at conferences, to multi-day, to courses, which are generally around a week long. And we recently, this year, published the first training paper on Bioconductor training. So it's in PLOS Computational Biology. The two first authors are Jenny Drenovich and Frederick Tan, who are both here at this conference. And it was led by the Bioconductor training chairs. So Charlotte Soneson, who spoke this morning, and Laurent Gatto. So that's a nice resource if you want to check out the trainings that we have available. Okay, and something we had been doing for the last couple of years since I joined as community manager was we had started collaboration, or Bioconductor had started collaboration with the Carpentries. So hands up if you know of the Carpentries. I'm going to assume a lot of people do. Okay, I see some people don't. So they're a global organization that provide foundational data analysis skills. So for introduction to R, Python, Unix, and now HPC. So we collaborated with them to enable people to get trained through their instructor certification program. So, and thanks to a grant from CCI, we were able to fund, it was 30, but we had 31 in the end, instructors trained around the world to become certified in teaching carpentries workshops. And as part of that, we also developed some lessons. So there's Bioconductor Carpentry now lesson in Introduction to R and Bioconductor, also on bulk RNA sequencing analysis, also more recently single cell. And so this has been a really nice program that's enabled us to train people around the world. And the idea here then is to help them to be able to deliver workshops and build skills locally and within their communities. So there's a blog post on the Bioconductor blog that you can have a read if you want more details on that, summarizing our 2 years on that program. But something, again, we had noticed was we delivered workshops. There was about 28 workshops delivered over the 2 years that were using that material and by instructors who were carpentry certified. But you can see again, there's no green is where workshops have taken place in person, and grey, Africa is grey. So we hadn't been able to deliver any workshops in person there. So we were lucky to get another CCI grant. So last year, which is to deliver these in-person workshops in Africa, in East and West Africa. And we were doing this in collaboration with some African bioinformatics PIs. So Trushar, Zedias, and Umar. And I have to acknowledge and thank Mike Love here because he was the instigator of connecting Trushar, who really wanted to bring workshops to Africa with me and the Bioconductor Community Advisory Board. So thanks for making that happen. So that's the blog post on the grants that we got funded through CCI. And then this led to our very first in-person bioconductor workshop that took place in Kenya in March. So it was a week long. It was held at Trushar's Institute, so the International Institute of Tropical Agriculture in Nairobi. A week-long course. So the first 2 days were our bioconductor carpentry introduction to R. The second 2 days were an RNA-seq, the bulk RNA-seq analysis, bioconductor carpentry lesson. And then the final day, the Friday, was where the learners could bring their own data to practice what they had learnt, or to use some of the example data sets we had. And we had a blend. We didn't want just to send international people there to teach. We wanted to make sure we were helping to build capacity locally. So we had both international and local carpentry certified instructors teaching at this workshop, 5 in all. We had huge demand for places. So over 145 applications for the 32 places that we had. So it was a tough selection, but we got a really good bunch shown in this photo here. So along with the instructors, and we had a mix. This bar chart is showing most of them were beginners, had never used R or just barely. And then a few of them, some of them were intermediate and just wanted the chance of being able to go to an in-person course, focusing on RNA sequencing analysis. And something unique we did here was, because we had some people at different levels, we adapted to how they were learning. So after we finished the intro course, before we started the RNA-seq, we gave people an opportunity to do a deeper revision going through the intro to R if they wanted. So as a parallel stream, if they wanted to spend more time getting foundations while the rest started with the RNA sequencing. That's mentioned in our blog post here that has more details on what we did and the course, and that worked really well. So people, about a third of the course actually ended up going into that stream then, so they could join on day 3 or day 4 or day 5, and they found that really great. So this is just a quote from one of the learners saying they really liked this special room or clinic or stream, we called it. They'd no experience with R, and so they felt much more comfortable with using R at the end. And we had lots more nice feedback. The learners themselves just started posting how much they were enjoying the course on LinkedIn, which was very nice. And yep, so a bonus outcome that came out of that was us instructors and organisers who were sitting around having dinner. We're talking about, there's this company called Phasalia that offers courses. Some of the bioconductor people were teaching. So we thought, wouldn't it be nice if we tried to have a collaboration with them where they would offer free places for, say, these learners in Africa who would never be able to pay the cost of attending the course. And so that's just set up and launched in the last week or so. And so that's a nice way, because the learners really wanted to be able to learn more after that week. So the next steps then are, we're planning the in-person course in West Africa. We've got 3 groups interested to host it. And we're also translating the material into French, because there's French-speaking regions around West Africa. And we'll also be then, after that, we'll be holding advanced workshops on specialised topics, probably online, and we'll be seeking instructors who would be willing to teach. Those might just be a couple of hours. So if anyone's interested in that, please keep an eye out for that call. And so, yeah, if you want to engage with us, we'd love anyone to join with what we're doing. You can find all the links of how to meet with us on training.bioconductor.org, monthly meetings. Anyone is welcome to drop in and join. And yeah, we would love any of your comments or feedback on what we're doing. So finally, thank you for listening to me, and thanks to these people, brilliant group, for organising these workshops. And yeah, talk to me if you're interested in this. Thanks.
Great timing. There's time for a question.
Oh, yeah.
Hi, I'm Michelle Savage, and I work on the Galaxy team. You know, that's really amazing, that everything that you're doing. And I'm wondering, what role did, because Africa is so mobile first, what role did your mobile access or applications play in your training in these locations?
Do you mean how did they find out about it, or find out about the course?
I mean,
or
the training itself, was it, did it utilize any mobile features that you guys offer, or was it all desktop based, I guess?
Oh, by mobile features, you mean like, can you give me? Oh, like mobile phones?
Yes, sorry, mobile phones or tablets.
Oh,
versus-
it was all laptop. Yeah. So participants installed the packages. So we gave them the instructions on how to install before the course start. And actually they all came prepared. We helped a few of them with install, but yeah, it was laptop based.
Great. Thank you.
Thanks, Mike.
Sorry.
So first of all, I would like to thanks. So before presenting our project, basically, I would like to first acknowledge the people that are working hard and really making it happen. Starting by Carole, our project manager, as well as Charles, who got a poster yesterday, but also more familiar faces like Enis and John from Johns Hopkins, as well as Nuwan from Melbourne. With who we team up for the ACCP project, on which I will talk about in a few slides. So before getting into the details of our implementation, I want to quickly introduce the ecosystem in which we are involving in Canada, which is a country up north. So for about 20 years, we had this Federation of Compute Canada that eventually evolved into the DRAC, so the Digital Research Alliance of Canada, basically for which we have a few national sites that are really hosting few resource. So and yeah, UDS is just here in Canada, so about 7 hours drive from New York. Yeah, so basically in the complete infrastructure in the total, so we have a pretty fair amount of resource, and by default every PI can have access to default allocation to get access to these results for free. And this is the amount, and on top of that, there's also the cloud resource that are on which we can tap for our project. And yeah, so among the biggest cloud resource in Canada is this Arbutus cloud, but we also have the Calcul Quebec also have this Beluga cloud, and UseGalaxy Canada is installed and managed in this facility in Montreal, managed by Calcul Quebec. Yeah, so since we started from scratch to implement the UseGalaxy and to ease our life basically, and based on recommendations from colleagues in the UseGalaxy Star, so we decided to only install tools that are running in biocontainers. So we currently have like about 500 vCPUs, 3 terabytes of RAM, and another terabyte of storage. So far, the mandatory single sign-on authentication is required through CI logon, so linked to the institution of the user. Upon the authentication, so each Canadian users receive 500 gigabytes of storage, international 250, and all the others only receive 5 gigabytes of storage with limited number of jobs, concurrent jobs that can run. And we do have a storage backup on the tape archive. So what about the usage? So because we still have limited resources, we haven't been really aggressive on the advertising, but still we just crossed the 600 users with more than 91,000 jobs that were run. And to answer a question that I had the other day about what is the profile of the users that we have, so we look at the top. Among the top tools that are used, and the answer is mainly life sciences. So the metagenomic, RNA-seq, and ChIP-seq, for instance. So this is the implementation that we did for the UseGalaxy Canada based on the recommendation from our colleagues. And at first we initially, we're about to spawn a pulsar node on the Arbutus on which there are some GPUs, but then arrived the ACCP project that I will talk more about in the next slide. So this is this Alliance Cloud pilot project basically allowing to spawn some pulsar nodes on commercial clouds. And for that, we generated, we developed this pulsar registry that we called. So ACCP stands for the Alliance Cloud Connect project. That was like about 6-month project, like kind of a bit crazy thing. And yeah, as I mentioned, so we team up with Nuwan, Enis, and John and we proposed 2 modes basically. The first mode was the bring your own compute and basically provision additional compute capacity, basically leverage the pulsar to run jobs on different cloud providers. And there was this other second mode, the launch your own Galaxy, standalone private data using Kubernetes and help infrastructure as a code on behind. So when we submitted the abstract for today, so the sign was that this pilot project should be reconduct and eventually become like a project in production, but unfortunately, we it just ended up in May. So, but still, so we learned some interesting lessons from that. So by integrating flexible, scalable cloud resource into the existing Galaxy framework, it provides researchers with new levels of control and capacity. This capacity charts path forward for decentralized, user-driven resource scaling across the global UseGalaxy star. And we are currently exploring options to connect to alternative, more resource providers, including, so talking briefly about this. So the plan is to use something that was developed in Calco, Quebec called the Magic Castle, which is a way to basically spawn some HPC. And it was developed, one reason for which it was developed to really ease the teach and the learning in HPC. So it's really simple to use. It can auto scale the infrastructure, and it's also cloud agnostic. So on the back end, it's using the Terraform and with a bit of automation, so we can then use this to spawn some pulsars in different sources. And then the next step is to basically link the UseGalaxy Canada with something similar to what we developed the Pulsar registry. So during a really interesting talk over the last days and a really nice discussion yesterday. So there's also this the TPV broker that was presented by Paul and developed with other colleagues. And we were just discussing about how we can integrate all this into our infrastructure, basically with the idea of providing the bring your own compute. So another thing that we really want to do over the next the next months is also to integrate Globus, which is a way to really ease the file transfer between, let's say, because I forgot to mention, but basically in the compute in the DRAC, so the Alliance, so we already have a license such that every researchers can already use Globus to ease the file transfer, which is also the case in most of the sequencing centers. So most of the files are transferred that way. So having a way to use to develop Globus interfaces will also be really useful for us. So basically, so the roadmap, basically, we will, of course, update to V25 soon. So we will be more active on user recruitment, improve the storage scalability performance. As I mentioned, add the Globus access to ease the import-export from Galaxy. Also add the Magic Castle to support pulsars to bring your own compute, and also a few more other improvements. So that's it.
So we have time for 2 questions, maybe. Oh, one right in the front.
Yeah, very cool talk. Thank you. So I was wondering if you could talk about the auto scaling on like a traditional shared cluster. Like, is that something that, you know, is that sort of used widely? Like, do you have, you know, maybe like a front end VM, and then you have jobs, Slurm jobs scheduled behind the scenes, and then Like, is that
Yeah,
the way to
so
use
I'm really not the technical person to be, I mean, the best place to answer. So Charles, which is in the, yeah, just over there. So he can answer over on top. So you can answer right away. But basically, the main idea is basically it's using also, so Slurm, basically when the nodes are not used, so they will just be down. And when there's a new request, so then Slurm will just, and they will spawn. And the idea here is not to really have to decide the size of the nodes, and we can just adopt based on the demand, basically. That's the main idea.
Is there another question? Okay, if not, we go on to the next speaker. Thank you.
Thank you. So yeah, my name is Subina Mehta. I'm part of the Galaxy for Proteomics team at the University of Minnesota. Apologies for the very long title. Every time I come to GCC, I believe that I should shorten the title, but it gets longer and longer every time. But the gist of it is basically I'm going to talk about mass spectrometry centered to multi-omics, and also how we are leveraging the Galaxy training materials to reach mass audience. So I'm not going to go into detail about what is mass spectrometry or what is proteomics, just basically want to emphasize how mass spectrometry has like revolutionized how we analyze like biological questions, how we understand proteins. And it's a very high throughput, sensitive, highly adaptable to all kinds of biological questions. And it is very crucial to the field of multi-omics, where we integrate proteogenomics and metaproteomics. Just to go into the flow of information in the multi-omics cascade, the genomics basically tells us what appears to be, like what can be in the sample. The transcriptomics is telling you what appears to be happening in the sample. The proteomics tells you what makes it happen, as well as what is expressed in the sample. And metabolomics is telling you what has happened and is happening in your sample. So basically, integrating these multi-omics data is something we need to understand what exactly is going on in your data set. And as this talk is mostly focused on mass spec, so I'm going to talk about mass spec based. But if you look at proteogenomics, it's basically telling you how we can identify like novel peptides, novel proteins, and or variant proteins, variant peptides that are present in your sample, which are different to the reference. And which is very important when you're doing cancer research or personalized medicine. With respect to metaproteomics, it's telling you what kind of profile is present, like what kind of complex microbiome is present in your sample, and not just at the sequencing level, but also functionally, what are they doing in the environment that they're living in, whether it's like environmental biology or whether it's like in the human body, like what they're doing. I think that's important for us to know. But like every omics, like genomics or proteomics, there are challenges that we face too. With the new instruments that come in, the data size increases, there's a lot of volume, there's a lot of different kinds of data. And the biggest challenge we face is the computational power of all of these tools. And also, the issue is like standardizing the analysis across labs, across users is kind of getting tougher. And also the new researchers lack the computational ability to use the existing tools efficiently. So our solution was, okay, why not use Galaxy? And how do we reach mass audience? The Galaxy Training Network. That's where this comes in. And as you can see, GTN offers a lot of different topics. And as you can see in the life science section, we have about 41 proteomics-based tutorials. And what this does is it's not just open source or web-based. It's like a community curated training, which is designed for both beginners as well as advanced users. And the GTN is also doing a lot of like sharing of information via its platform, like Galaxy platform, via Smorgasbord or even Galaxy Training Academy, to reach mass audience, to reach new users, and to expose them to the Galaxy platform. There are 41 tutorials, as I showed before, that are done in the field of proteomics. And Galaxy P, like our team, has contributed to about 20 of them. And because our focus is mainly proteomics, we work with the GTN developers to help build these workflows. And in the fields of like genomics, transcriptomics, neoantigen prediction, to metaproteomics or microbiome studies with both environmental as well as clinical samples. Now, GTN tutorials are not just walkthroughs. They are most of them are using real data sets and answering real biological questions. They are designed to be modular so that any user who reads through or goes through is confident about the step-by-step analysis. And also because it is structured and well supported, even users who have no kind of coding experience can use this. And also we provide like sample data sets so that you can get your hands wet into using Galaxy platform as well as the tools that we have. So basically, I just want to say is that it's not just the context that the GTN is giving. It's giving you basically all the recommended tools or different kinds of workflows, or even it's telling you what are the alternatives that you can use. And it's also giving you supposed parameters that you can use for doing your data analysis. And at the end of the day, every researcher wants to interpret their data. So for that also, we provide you with like different alternatives or visualizations that you can use to visualize your data, interpret the data, manipulate your data. So all these information is provided by the GTN so that you can do your preliminary research. And at the end of the day, what is this doing? It's all promoting community adoption. And that is what is our end goal. If you look at this map, these are some of the metaproteomics-based workshops that we have conducted over the years. And all of these are using the GTN capabilities. We just conducted a workshop like 10 minutes ago from 2:30 to 3:30, and we use the GTN quite a lot. The learning pathways in the GTN, which actually walks you through all the different workflows that we have developed, and which is kind of helpful that the users can take it home and then rethink about what they did in the workshops and also follow through and also provide us feedback. So kind of all these global outreaches that we have done, we have gotten a lot of feedback regarding our tutorials, which has helped us make it better. So in summary, I think I've talked way too fast. I have a lot of time. I just wanted to say that Galaxy is beyond tool usage. It's not just about developing tools or developing workflows or different kinds of platforms. Our main goal, at least in the Galaxy P team, is like to equip users to have confidence in how to interpret the data. Also, this is made possible by the Galaxy team all across the globe and the GTN. And we have tried to make the field of proteomics more accessible and multi-omics also more accessible to new users. And we believe like with the growing suite of tools that we have, the growing suite of tutorials that we have, and with all the community contributions that we do, we would at least be able to make these wide range of scientists across the world, like Maria is doing with Bioconductor. At least we feel like we can do that with Galaxy, to engage, and we want them to benefit by using these kind of powerful tools. So with that, I would like to acknowledge Galaxy P team, the Supercomputing Institute, UseGalaxy.org, and our collaborators across the world and our funding agencies for providing us with the opportunity to do this. Thank you, and please let me know if you have any questions.
Any questions?
I mean, I know it's all about training, so
I-
it's not.
I have one ready to go. So one thing we like try to work on and be better is like providing spots where you ask the person who's working through the training material, like, stop and what do you think about this? Like, so for proteomics, what are the big questions where you say, okay, you can't just keep going? Like what, you stop and you have to look and make a decision.
So for us, we create different modules. So when we create a workflow, when we put it in the training network, we cut the workflow into various steps so that after every step, you look at what the results are. If you're not happy with it and you want to redo it, you can interfere at that time, because at the end of the day, there is human intervention that is needed. So we do step-by-step modules. So if you go to Galaxy Training Network, there's learning pathways that we are developing so that you can go step by step and look at workflows by workflows and then analyze end results of each of them. So that kind of helps.
Perfect. Thank you.
All right. Thank you so much, everybody. Good afternoon. I'm Natalie Kucher. I'm a project manager at Johns Hopkins University. Today, I'm going to talk to you about training and education in the cloud with the NHGRI AnVIL, which some of you might have gotten a taste of at our workshop just before this. I don't need to tell this audience much about the growth of genomics data, the growth of biomedical research data. Subina just highlighted that as well. This creates a need for tools to better share and manage data with collaborators, also access data, computing resources and tools to be able to interact with and analyze this data. So that's a large motivation for AnVIL, which is the Analysis, Visualization, and Informatics Lab Space. This is an ecosystem that supports researchers who are accessing protected data sets. So AnVIL hosts over 7 petabytes of human genomic data sets that are generated by the NHGRI funded consortia. You can also bring your own data to AnVIL as well. So you can combine those data sets with yours in research, and you could also upload any data you'd like as well. The boundary is really to protect the human genomic data sets. AnVIL also provides a lot of compute resources as well. We're built on commercial cloud platform GCP, so you're able to use as many resources as you need for your analysis. And AnVIL integrates great bioinformatics tools like Bioconductor and Galaxy that were here for this conference, as well as Jupyter notebooks and workflows. So you're able to create your own or import those that are available in DocStore for workflows. And AnVIL also supports a vibrant community of researchers. So we have a great support forum where folks can post their questions and share answers and get help. And then AnVIL also really supports sharing of your data with collaborators who are outside of your institution with specific fine-grained access controls that Stephen mentioned if you're working with protected data. So this provides a nice ecosystem where you have all the data that you need, tools and compute that you need to be able to work at scale. So now that we talked a little bit about AnVIL, a couple years ago, the AnVIL outreach team wanted to get a better sense of who our community is. Where are people doing their compute? What kind of tools are folks using? And where can we put our focus in terms of developing training materials for our audience? So we launched this poll last year. I'm only going to highlight a couple of the responses that we've got, but our current poll is still live. So if you'd like to fill that out, we'd love to hear what you're up to. The QR code's in the corner. The first result that I'll talk to you about is where do users currently run their analyses? So maybe not surprisingly, most of our respondents said that they work on their personal computer or institutional HPC. Based on the traditional model of data science, a lot of folks do work pretty locally, but we're seeing a lot of our respondents, especially on AnVIL, who are Google Cloud Platform users. So we're wanting to build more materials for folks to gain familiarity with working in the cloud. And the other result that we saw was trying to understand which tools folks tend to use. So there's a pretty widespread with a lot of familiarity with tools like command line, R in RStudio, Python, notebooks, and Bioconductor. So AnVIL is a great environment for folks who have a spread of tools that they'd like to use. So now shifting focus into how do we do training in the cloud? You might be familiar with a number of training resources or versions of the tools that you use that are in the cloud. And here, there are a lot of advantages, like having identical learner environments where tools and environments are ready to go, and students don't need to configure a lot of that before they get to the workshop or like the first 30 minutes. And a lot of these resources also have really strong community support and training materials ready to go. But with AnVIL, what you're able to do is bridge these training experiences with an experience working in an environment that researchers are using protected human genomics data sets. Because it is a secure platform for these controlled access data, whatever familiarity you have working on open access data sets or tools, that translates really well to working on more of these protected data. You also have customization by being able to install software through Conda and Docker. And then you also have the flexibility working on a commercial cloud platform to pay to scale your resources. So you're not in a queue, and you're able to initialize an environment at whatever size you need. AnVIL also, like I mentioned, has a lot of collaboration tools where you're able to work with folks that are at other institutions or other folks who have access to the data that you're working on. And you're also able to publish and share the results of your work by sharing a link to your workspace, which a number of publications have done, and we're really excited to see that as well. So in the last couple of minutes, I do want to highlight some of the educational materials that have been developed by our outreach team. On the left side is a QR code to the AnVIL collection. This is where we list out all of the materials that we've developed. And I'd like to highlight these examples as those that use Bioconductor with an IC example that was mentioned in our keynote this morning, as well as developing an R data package for a project that we're working on that's collecting soil metagenomics data and environmental data as well. We also have an activity analyzing the SARS genome with Galaxy on AnVIL, and an RNA-seq mini-cure that utilizes Bioconductor. So these resources vary from being able to run them in like 30 minutes on your own to learn how to use AnVIL, or examples like the SARS activity, you can set that up and have your students run that in a class, or the RNA-seq mini-cure that could take place over the length of a course. All of our materials are open source and modular. So many of these come with lecture videos that are ready to go. These have access. You have access to the Google Slides. So if in your course you have other context that you want to include or specific details you'd like to dive into, you can add that to the video as well. But they're also rendered in the book so students can watch them on their own. And there are student activity guides where the students can go through the exercises and answer all of those questions, like was mentioned in the last talk about, oh, what variant do you see, or how do you interpret this visualization? And finally, here, I do want to really echo Subina's comments about the Galaxy Training Network. This has been an excellent resource with a really strong precedent for having open training materials. So we're working to also add our Galaxy tutorials to the GTN as well, just to promote as much visibility as possible across these resources. And my final slide here, I do want to highlight that we do have a resource for instructors too. So we have a book called the AnVIL Instructor Guide. And here you will have all of the checklists, which this is a screenshot of, that even we use for the workshops that we host on AnVIL. So everything from how to set up your accounts, how to set up your students and your TAs, how to manage billing and make sure that all the environments are shut down at the end of the workshop. So I would really highlight this resource as a place for you as an instructor to get started to bring AnVIL into the classroom. So with that, I'd like to just thank the AnVIL outreach team, especially for developing these materials. We've been working really closely with a number of collaborators who are not listed here, who developed things like the RNA-seq mini-cure and the SARS activity that we've lifted to AnVIL, and are really grateful for their work with us. A couple of places you can find us is we have monthly AnVIL demos. So if you ever have questions, feel free to jump on and we'll be delighted to talk with you. the 24/7 support forum, which is modeled after Galaxy support forum. And we have a community conference coming up in September where we'd love to see you as well. Thanks so much.
We have time for a question. Oh.
Mentioned some access in classrooms. I'm wondering who are the students that are most interested in using AnVIL? Are these medical students? Are they undergraduates? What's your target audience for that?
That's a great question. We're trying to cast a wide net, but we've been working really closely with this group called the Genomic Data Science Community Network. These are primarily educators at undergraduate institutions across the country where they don't have HPC and they don't have a lot of computing resources. So that's where we've seen a lot of interest and have been focusing a lot of our materials to at least meet the needs of those students who are really interested to get involved in cloud computing and genomic data science, so that that's a great foundation for anyone who's coming to us as well.
Thanks.
Great. Let's thank our speaker again.
Hi, everyone, and thanks for being here today, listening to me talk about how I use Galaxy to empower research in developing communities, because it's an honor to be around people who know how it works, but I'm here on the side of people who use it and do not know how exactly things work, but we are thankful that this tool exists. I'm a visiting professor at a Brazilian state university at the Bahia state. And so just a little bit of context before you all will understand how this has been such a useful tool for us. So the university is 49 years old, and it's not like a very old university, but it's one of the oldest in the area. So it does welcome students from all over the world, but over 90% of them are local from Feira de Santana, which is the city it stands, or other cities in Bahia, which are near or maybe more far away. And here I just bring a little bit of context of why having a laptop for each student is not that easy in our case. So a basic laptop that would allow for maybe amplicon analysis would cost around 3,000 Brazilian reais, which is like twice the per capita income in my state. So this is one of the reasons why I believe these tools make it so much easier for us to study microbiomes and the genomics as a whole. So the area where my university stays is a transition area between the littoral biome, which is the Atlantic rainforest. Maybe some of you have heard a little bit about it, but it has like this huge, massive semi-arid region called the Caatinga, which is like the white forest in the natives' language. And it stays like in the first slide, or like a white forest for maybe 9 to 10 months a year. And it's not very well studied yet because we stay in the poorest region of the country. So we still don't have like that much power to study everything about the biodiversity. Although we do have like a lot of information about fauna and flora, the microbiome, which is what I'm interested in, it's still lacking a lot of studies. Keep in mind that this could fit like 2 German territories and a little more. So it's huge. So that means it's not enough. In this sense, so it is also a unique biome. It's the only biome that is unique to Brazil. You won't find Caatinga anywhere else. Differently from the Amazon, which we share with other countries and everything. So for this reason, we believe that studying the microbiomes in the area is definitely a very important thing, because this biome has been altered by over 80%, but because it's semi-arid, it means it's very prone to desertification, and this has been identified before. So it's just like getting lost. This is the reason why at UFs, we have been started to think, how can we help with the studies? How can we bring more information about the biodiversity if we lack a lot of the infrastructure that it usually takes? So one of the things that we don't have an HPC at the university, or that means my undergrad students don't really get exposed to the bioinformatics and genomics as much. Although the biology course is actually one of the oldest in the institution, we still lack that. So we don't have bioinformaticians that much. So how can we do that? So it's one of the things that I think it's wonderful about being here. It's showing maybe you guys don't know how much it is important for us on the other side, because this is what I have been using to get students to learn about genomics and to get a sense of what bioinformatics is. Because in the biology course, we also don't teach them how to program. So using the platform that you don't have to do programming and that you have like a lot of space available, I did have students using the same computer, each one like a time and doing their things. And when they do have a computer, it's usually a basic one for studying, for reading, and not capable of like the performance that we need to. Also, you cannot break anything if it's not running locally. And this is something that really scares my students. Like, we're doing this and that. Sometimes they come to me and say, I didn't do what you asked for because I didn't know where to press. And I was like, it's not even on the computer. We can just start fresh. But it happens. So not getting risking to break anything, break your own computer or whatever, that's also huge. Maybe it's something that we don't think of that much, but it really makes a difference. So the idea was to build some capacity through teaching using Galaxy. So in this case, I ran this workshop, like it's bioinformatics for environmental microbiomes in Portuguese. It's been 2 editions so far. It really is something very informal. I will just like make it available and anyone can just join. There is never like too many people, but always a nice group. So we will be focusing on the concepts and step by step. Like I will tell them what we are doing and why we are doing that. And they won't have to worry about programming or anything else like that, because I know my students don't know how to program, or maybe most of them. So, and we also usually, we use locally publicly available data. So we try to use some data from Bahia, probably mostly from other biomes. But we use that and we go through like the pre-processing and analyzing and discuss what we are doing. So I can teach what we're doing without having the stress of having them programming or trying to understand the commands. And in this case, this last session was in January. It was the summer vacations, but I still had people interested. And I had mostly graduate students, but also some fellow professors wanted to be more like up to date where biology is going, because we all need to do that. So we went through the time to pipeline, which is available through the Galaxy platform. And we covered like through alpha diversity, beta diversity, taxonomies, and some differential abundances. And we discussed why we were doing that. And so after all, the students showed some interest in expanding the analysis further. So I got to show them what R looks like when we are using it for microbiome analysis. And they got to take a look at how different these experiences. At some point, you don't have to do programming. And on the other hand, sometimes you can use your own laptop. And maybe there are laptops after all the pre-processing, we can start running. So this is where I say, so you can use some of the Bioconductor packages here, and we can move on, and you can be more independent if you feel like it, but like after going to a certain point. And at the end, we have identified some greater issues. So there is the language barrier. So maybe a tutorial in Portuguese would be helpful. So they would feel like more independent. And maybe because we have the problem with access to personal computers, online workshops are always a best option because sometimes they will share computers with someone else. And also we need more time. 2 weeks was not enough. Like we did have a lot of problem, but this is, we are all learning. So, and the next steps is like, we know there is the interest and more languages would be more helpful maybe for like reaching to more people. Not risking to lose or damage a computer is a great deal. I learned that. And we are trying to build a longer program. So we have like a bioinformatics, a curricularization. So we are working towards that. And this was my presentation. I want to thank you, my fellow colleagues, Peter Spielman, Gabriel Perron, and Thais Bonfim, who have been always helping me through this, and some of the students that were still online when I decided to take the picture. Thank you.
Hi, everybody. Yes, I work in a Science Institute from Adelaide, and we work a lot with the Human Cell Atlas, which is this amazing resource repository of more than 100 million cells across tissues, cell types, and the human population. So we are in a situation where, as I said, we have available 100 million cell types. But we are just in an exponential increase where in the next 5 years, there are 2 major projects where there is 1 billion cell atlas from CZI and 10X and the 5 billion atlas from the Broad and Illumina. So for example, we survey this data to an example to model how the immune system is heterogeneous across the population. But you can imagine you could use this data to map your experiment to a very comprehensive, healthy reference of the population specific to organs or a disease specific. So it's very important that we are ready to be able to extract knowledge from this wealth of information. So a barrier to that is that you have to spend weeks and months to harmonize this data from this repository, and not all laboratories can afford that. So we wanted to create a quality control and annotation layer so you can analyze, you can do data from analysis in a very efficient manner. So we started grabbing the data from these cell by gene repositories served by the Census API. And these include, at the moment, 50 million cells that we selected across tissues. And we have quality control, the counts. This is gene expression data. So it's count gene per cell, is a big matrix, if you can imagine. And we have provide normalization for these counts. But not only, we also provide quality control for the cells. So we do empty cell classification, doublets, and dead alive cells classification. But also we improve the metadata of those cells, for example, with cell typing, consensus, and impute missing labels such as sex and ethnicity. And the user can decide to use our API, which is called Cell Nexus, and filter the cells of interest, let's say T cells across the human body for older individuals, healthy individuals, and we deploy directly a data container such as single-cell experiments, rat, and data, and so on, that you can start the analysis with straight away. If you want to use the original data with the Census API, you can still map our enhanced metadata annotation onto the original data with cross-linking of one ID. And why not? This resource could be also helpful for the Galaxy environment where huge data set could be mapped onto pretty easily. So who was on Bioconductor maybe 1 year or more ago? The difference with the package I showed the last time, which is called Curated Atlas Query, we learned from that experience and we created this Cell Nexus, which will replace that. So what's the difference on that? Well, first of all, we'll remake the whole reproducible pipeline, scalable, that is able to get the raw data and produce what we are producing. And this is use R targets, which is a workflow system on the HPC. And we are basing our file system on H5AD. So it's made us able to provide API for R and Python. And of course, we adapted to the latest version of the data, as well as I will show you now, we provide different resolution representation of the data set. Okay, this is quite low resolution here, but why quality control is important is a small text, but I will just point you to the right place. In this bar plot, we show that depending on the technology, there is up to 15% of cells that we label as low quality. So this would save you weeks and months to do this quality control at scale, just to select the cells you want from different data sets. So you can start from a high quality data set. There are very strange count distribution, which go from here -200 to 200. So it's not easy to interpret. So we kind of harmonize that with some criteria. And as well, again, text is very small, but in the middle plot, 50% of samples have missing ethnicity annotation. So it's the very lack of potential in there. And we try to impute those labels for you. We have the resource that is on the cloud, but we provide the API as well. So you can see on top for R and Python on the bottom, but basically it's very simple. It works with kind of natural language scripting. You call get metadata, so the metadata in a Parquet format will be downloaded on your computer, and you can explore and filter 50 million cells on your laptop. Is very efficient in that sense. And then once you know the content of the data you want and you filter the cells you want, you can call get single cell experiment and data what you want. And the files that you need, just the one that you need, will be downloaded and put together, packaged in these data containers. And you can see, again, the text is pretty small and for some reason, not the resolution that I was expecting. But you can see you can use tidyverse to do all these operations. So it's quite, as I show you in the lighting talk, it's quite natural to understand what's going on. You can filter, in this case, different criteria. And again, you get your data container ready for you. Same thing for Python. There is a quite SQL-like filter operation. And so the two datasets platform are quite consistent. We provide a web app, so you can do all these kind of point and click just to go quickly through it. But most importantly, in order to really democratize this, we can imagine that as we train linear models, we will train foundational models in the future of the data we want. So very few laboratories will be able to afford a 1 billion cell model or atlas. So we think that providing meta cell representation, so you join the closest 2 cell, 4 cell, 8 cell. So in a kind of a spectrum of aggregation, you can choose the aggregation you can afford to analyze your million cell or even more data set. And this will able to really democratize the use of these resources. And we tested this hypothesis. So we say, okay, we want to build the foundational models using GeneFormer here. And we test a single cell against meta cell representations. And you can see there that we have, well, again, number of cells on the x-axis and time. So it scales quite linearly. A kind of a nice accident that the bigger model we could not finish training. So there are checkpoints after a few epochs. But you can see that the time is the same. So we cannot compare the performances at the capped resources using more trained aggregated representation versus the other way around. And we train against few tasks that this GeneFormer paper proposes. One thing is that if we give a data set, this is from the GeneFormer paper, and we ask which embedding this model produces, all the embedding across aggregation, representation, they look similar, so they are not corrupted. So this is a quite nice qualitative observation to start with. Another task is prediction of gene dosage sensitivity. And just to make the long story short, you can see here the area under the curve is quite flat, which is good to see. And interestingly, for the big models that took the same time as the middle representation metacell, they perform worse. So if we train for the same time, is much better to train an aggregated representation than a single cell. And you can imagine it's the same thing. We want to train aggregated representation rather than subset data because you don't have the power to do it. And again, just to show another task, this is cell type classification on a cardiomyopathy dataset. And you can see the confusion matrix on the diagonals that look quite similar. And the accuracy is shown in that plot. Basically, again, we have quite a flat performance, and even the single cell, so the most right, that has been trained with the same timing as the middle one, perform worse. Again, tell you at the same resources, the aggregate is much more efficient way to learn from these models. That's it. I have to thank many people because this work comes from a long history. So the Tidiomics community, as well as my institute and many others. And thanks.
Great. There's time for one question. I can.
Hey, Stefano, great talk, super useful resource. The ARC Institute came recently out with the Virtual Cell Atlas, which contains of SC Basecamp and this Tahoe data set, which is like 230 million cells exceeding CellX gene. Can you imagine providing something similar in Bioconductor for these resources?
Yeah, definitely. So the thing we do is very scalable. It could be even deployed and hosted in different places because it's file system based. I'm curious to learn more about that. I did not understand well what that is about, but definitely if there are even bigger resources, definitely we are interested in.
So they're doing something similar like CellX gene, but on a bigger scale, and they're employing kind of like AI to curate and annotate the metadata, but they're collecting basically systematically from the
who's the institution?
The ARC Institute.
ARC Institute.
So
Okay.
Research
Great.
Institute in the US.
Amazing. Yeah, let's talk about that. Very interesting.
Great. Let's thank all the speakers for our session.